Welcome to P3, I'm Randall Mark. This is the show that explores the fascinating people, places, and perspectives that make up our world. On today's show, I get a chance to explore some groundbreaking medical research being done in Israel. I meet up with a spinal cord researcher that's teaching paralyzed to walk again. This is P3. Stem cell research is controversial because the best source for these cells is human fetal tissue, and using them destroys the embryo. Dr. Heinz Cedar of the Institute for Medical Research Israel, Canada at the Hebrew University of Jerusalem is one of the world's foremost authorities on DNA and genetics. He explained to me the intricate workings of the science behind stem cells and what has to be done to artificially replicate human body parts in a lab. Professor, we're here at Hebrew University, and you were doing some cutting-edge research on the issue of stem cells. And I know that in the past I've talked to you about how the cell regulates, how it gives information out, tells eye color, or tells the cell to be a hand or skin tissue. Explain the connection between that research and what we're discovering about stem cells.
So there's a tremendous interest in stem cells today. Basically, a stem cell comes from the very, very early embryo. In the very, very early embryo, you have all the genetic information, but the embryo hasn't decided yet or hasn't set the switches to which genes are going to be on and which ones are going to be off in order to develop the embryo. So these cells sort of have a lot of potential.
Pluripotent.
They're pluripotent.
Look at me knowing some words, huh?
You see that? Yeah. These cells are pluripotent. As they go on to become cells of the body, what we call somatic cells, these switches, like DNA methylation, are set. And now their information book has been annotated to express certain things and not express other things.
But if you could capture them before they are set, is the potential that those cells could be to grow other tissue?
If we knew how these switches are set, then we would be able to take a stem cell and program it so that it will set the switches to make a liver cell or a kidney cell or a nerve cell. And the opposite is also true, and that is that we can take somatic cells, cells of the body that have already been programmed, and now reset the switches and go back to a more stem cell or more pluripotent type of cell.
Do you see the possibility of you growing a human liver in a lab, a human kidney?
So we don't work on these problems. Our work is
the biological core level, the cellular level, yeah.
But the people who do this are using all sorts of materials and all sorts of compounds and chemicals to try and do this. And one of the principles behind this is that you try to use chemicals or inducers or substances that work on this process of methylation so that you get it to program the switches in the way that you want it to go. In cancer, we know that one of the things that happens is that the cell, which starts off as normal and has the normal regulatory modifications, it undergoes excess modification in places where it shouldn't be. And this causes big changes in what the cell does. And this, for instance, is responsible for why the cells grow rapidly and why they invade and why they can wander from one place to another. It's all because the gene regulation has been altered.
But if you could somehow control that regulation, you might be able to inhibit the cancer from growing.
That's right. So it's very possible that drugs that inhibit methylation can either be used for treating cancer, and even today it's used for treating certain types of tumors, or maybe it could even be used someday to prevent the development of cancer. Yeah.
That's an amazing finding to understand that at the biological level, the chemical level, to understand that process, but then to be able to possibly prevent that with some drug would be incredible for my children, my children's children.
That's the hope.
You've figured out these switches, how these switches are working
Exactly.
at the most basic cellular level.
Exactly.
It's an incredible finding. It's an incredible discovery that to figure out at that level, because the potential now to help people who need tissue, to regrow skin tissue, to regrow maybe even complex organs, is in our fingertips.
On that. And I think this is a general phenomenon that the more your research is aimed towards a basic fundamental understanding, that the information that you get will be much more useful for a wider range of problems than if you just work on research on the very, very end product.
Right.
And so this is the logic behind doing basic research.
Right. Well, thank you so much. It is absolutely fascinating where this potentially could go and how you could help so many people. Thank you so much for your research.
Coming up next on P3, I talk with a leading neuroscientist about the healing capabilities of the mind.